All right, today we're diving into a really groundbreaking scientific review all about GLP-1 and addiction. This is a paper that gives us some profound insights into how our own metabolism might just hold the key to a whole new frontier in treatment.
You know, for decades, addiction treatment has been, well, neurocentric. It's focused almost entirely on brain chemicals like dopamine. But the thing is, this approach has had pretty limited long-term success. So now, the whole paradigm is shifting. As you can see here, we're starting to look towards these metabolic signals coming from our gut, communicating through what's called the gut-brain axis, to help regulate the very motivation and reward circuits that fuel addiction. It's a huge shift in thinking.
Okay, so our journey through this paper is broken down into 5 parts. First, we'll start with the core challenge of addiction itself. Then we'll dive into the star of the show, this gut-to-brain messenger, GLP-1. From there, we'll see how it actually reshapes the brain's reward circuits, we'll analyze the crucial evidence, and finally, we'll look ahead and consider what the future holds for this entire approach.
Right. Let's start at the beginning. The core problem we're trying to solve. Addiction is a chronic, relapsing disorder. And let's be honest, despite our best efforts, conventional therapies often fall short. That's why we see such high relapse rates.
And the issue is, substance use disorders are just incredibly complex. By focusing so narrowly on neurotransmitters, a lot of traditional treatments miss this really critical link, the connection between our metabolic state and neuroplasticity, the brain's ability to physically rewire itself. And that oversight, that's exactly why we need new strategies.
And that brings us right to the key player in this whole new paradigm, a hormone called GLP-1. This is a molecule that operates at the very intersection of our metabolism and our mind.
So, what exactly is GLP-1? Well, it's a hormone that's produced mainly in our intestinal cells when we eat, but it's also made in the brainstem. And its job isn't just about regulating blood sugar. It acts as a powerful neuromodulator, which is basically a signaling molecule that directly influences key areas of the brain involved in reward.
Now, what's really fascinating is how GLP-1 actually gets its message to the brain. As this diagram shows, it uses 2 completely distinct routes. On one hand, it can activate the vagus nerve, which is like a direct neural highway from the gut right to the brainstem, or it can enter the bloodstream and travel directly to the brain that way. This dual pathway is what allows it to influence the central nervous system both quickly and broadly.
Okay, so we know it gets to the brain, but how does this process actually reshape the way our brain experiences reward? The answer lies in GLP-1's effect on a critical network known as the mesocortical limbic circuitry.
Think of this as a map of the brain's reward superhighway. What we see is that receptors for GLP-1 are found in really high concentrations in the ventral tegmental area, or VTA. That's the source of our dopamine. Also in the nucleus accumbens, which is central to motivation. And finally, the prefrontal cortex, which governs things like executive control and craving. By acting on all these regions, GLP-1 can directly blunt the dopamine firing that makes drugs and other rewards feel so powerful and compelling.
All right, let's zoom in even further, right down to the cellular level, because the mechanism here is incredibly sophisticated. Drugs that mimic GLP-1, like semaglutide, use a process called biased agonism. Now, what does that mean? As this diagram shows, they preferentially activate the G protein pathway, which is what leads to gene transcription and those important long-term neuroplastic changes. But at the same time, they limit the beta-arrestin pathway, and that's the one that can cause receptor desensitization and side effects. It's a really targeted and frankly elegant approach.
Okay, now for the real core of this review, the evidence. Does this theory actually hold up in practice? Let's break down the preclinical and the clinical findings for various substance use disorders.
First up, alcohol use disorder. Obviously, given its huge societal impact, finding effective new treatments here is a major priority. So let's see what the evidence shows for GLP-1.
The evidence here is really compelling, but it's also nuanced. In animal models, like you see on the left, GLP-1 agonists clearly reduce alcohol intake and suppress dopamine release. It's pretty clear-cut. But in humans, the results seem to depend on the drug and the patient. An older trial with a drug called exenatide found it reduced craving, but mainly in patients who also had obesity. However, a more recent trial with semaglutide, that's the exciting part, showed a robust reduction in alcohol intake with a large effect size, and this was regardless of a person's weight.
Now for nicotine addiction, this is where GLP-1 agonists present a really powerful dual benefit. Preclinical studies show they reduce nicotine self-administration and craving. But here's the crucial part. They also help attenuate the weight gain that so often comes with quitting smoking. And we know that's a major barrier for so many people trying to quit.
When it comes to psychostimulants like cocaine, the picture is a bit less clear. The animal studies are very promising. They showed that GLP-1 agonists can blunt that massive dopamine overflow you get from cocaine. However, a single low-dose human trial failed to reduce craving. So what's going on? The paper suggests a reason. Those long-term neuroplastic changes we talked about? They likely require chronic, consistent dosing, not just a one-off treatment. So this really highlights a key area for future research.
Okay, what about opioid use disorder? Here, GLP-1 seems to work by a slightly different mechanism. It actually targets the stress and anxiety circuits in a part of the brain called the central amygdala, and we know those are powerful triggers for relapse. The preclinical data is solid. It shows reduced heroin and fentanyl seeking. And the early clinical data from a trial with liraglutide is very promising, reporting about a 40% reduction in opioid craving.
And this slide really gets to the origin of these drugs and why they're so relevant to addiction. For food addiction and binge eating, GLP-1 is just an ideal therapeutic target. As you can see, it sits right at the intersection of metabolic regulation and the brain's reward circuitry. This lets it tackle the problem from both sides at once.
So this summary matrix gives us a really clear at-a-glance overview of where things stand. And here's the main takeaway. Across the board, from alcohol to opioids, the preclinical evidence is strong, no question. The clinical status, however, is more variable. It's definitely promising for alcohol and nicotine, but it's still mixed or very preliminary for cocaine and opioids. And this just highlights how critical trial design and dosing really are.
So, what does all of this mean for the future? Let's wrap things up and summarize the path forward, making sure we balance the potential benefits with the necessary precautions.
Now, a really critical consideration here is safety. Look, any drug that powerfully modulates the brain's reward system is going to carry potential risks. As this slide shows, the therapeutic efficacy absolutely has to be balanced against neuropsychiatric risks, like anhedonia. That's a blunting of pleasure from normal natural rewards, or other mood changes. And this makes careful screening for pre-existing conditions like depression absolutely essential. So the future here isn't a one-size-fits-all approach. It's all about precision medicine. Imagine tailoring GLP-1 therapy based on a patient's unique metabolism and even their vagal nerve function to get the maximum effect. This also points us toward the next generation of these drugs. We're talking dual and even triple agonists that act on multiple targets at once for a potentially even stronger, synergistic effect on reward.
All right, let's distill all of this down to 4 key take-home messages. First, GLP-1 is a central neuromodulator that goes way beyond just metabolism. It directly acts on the brain's core reward circuitry. Second, the mechanism isn't some simple dopamine blockade. It involves that sophisticated biased agonism, and it promotes long-term synaptic plasticity. Third, the clinical evidence right now is strongest for alcohol and smoking, and we've learned that dosing duration is absolutely critical for success. And finally, number 4, the future is all about precision medicine, targeting that gut-brain axis based on an individual patient's biology.
And all this leaves us with a final, really thought-provoking question. We are just beginning to understand the incredibly profound connection between our gut and our brain. So as we continue to bridge this gap between metabolism and the mind, could therapies that target this very axis truly represent the next great frontier in treating the complex challenge of addiction?
This explainer was based on the review Mechanisms of GLP-1 in Modulating Craving and Addiction, which was published in the journal Medical Sciences. Thanks so much for watching.